Hello, everyone. Welcome to Foresight's Biotech Group, sponsored by 100 Plus Capital. Today, we're really lucky to have Dr. CoCo Headley with us today from Stanford. His background is in microbiology, and he earned his Ph.D. in immunology and did his postdoc research in cardiovascular medicine at Stanford. His research explores the intersection of aging-associated mitochondrial dysfunction and immune regulation. So without further ado, today's talk is Bioengineering Mitochondria for Transplantation Therapy. We're so excited to have you here, CoCo. I think this has been a year in the works. So it's literally been a year, so we're really happy to have you on.
I appreciate it, and thank you for making the time for everyone for attending. Yeah, it has been, I guess, a bit of back and forth just because of the time. So yeah, I'm gonna try to yap for 30 to 40 minutes about the stuff that I've been working on. But before I start, I put my acknowledgments up front because it takes a village, and I want to thank everyone that's asked a question that made me ponder or help for this research. So mitochondrial dysfunction, to me, I kind of describe it as cellular engine failure. So in real life, if your check engine light comes on, you can probably go to the mechanic. If the engine is messed up, then you can probably go swap it out for a better one and keep using that car. And so similar thing happens in the body with your mitochondria. But when we talk about mitochondrial dysfunction, we don't really quite explain what is the engine failure. And so most people, I don't want to bore you with too much biochemistry, but the electron transport chain is how mitochondria produce ATP. Electrons get shuttled across. Electrons get shuttled across, and it builds a proton gradient in the intermembrane space that's then used by ATP synthase machinery to convert ADP into ATP. For whatever reason, you might have leakage of electrons, and typically that's been well characterized. Most of it comes from complexes 1 and 3 of the electron transport chain, and because you've got oxygen that's ever present, when you have electrons react with oxygen, you have reactive oxygen species formed. And so within the matrix of the mitochondria, you've got mitochondrial DNA, and we'll call this the wild type copy or the healthy copy. And reactive oxygen species, it's like a bar fight. They will pick a fight with it. They will damage anything that's around. And so some will hit the DNA, but you have these sort of bouncers or antioxidants like catalase, peroxidase mutase, and time peroxidants that help to counteract or counterbalance the ROS or oxidative stress that gets produced. You also have DNA polymerase that helps to repair any sort of damaged mitochondrial DNA. It typically has like a low error rate in its repair process, so you don't have a lot of mutations or deletions inserted during the repair process. But in instances of exuberant ROS, it's like a mosh pit or like a bar fight. So everything gets damaged. The antioxidants are supposed to help protect you, also become oxidized and damaged. The mitochondrial DNA gets oxidized. The enzymes that's supposed to, or the complex is supposed to help to repair mitochondrial DNA also gets damaged, and it becomes, the error rate increases, and you end up accumulating more mutated mitochondrial DNA. And so this is kind of like the perpetual loop of oxidative or reactive oxygen species production, damage to mitochondrial DNA that then influences the ability of your mitochondria to produce ATP, right? And this is the perpetual loop. And as we age, we talk about this in terms of heteroplasmy, which is sort of the ratio of healthy mitochondrial DNA to messed up mitochondrial DNA. Now, it's important to remember that mitochondrial DNA encodes about less than 1% of the whole mitochondrial genome, but all of the things encoded by mitochondrial DNA are indispensable. So if something goes wrong, you have dysfunction form. And so this is, as you get older, because you start to accumulate more mutated mitochondrial DNA, the expression of irregular proteins that are supposed to help with that help with assembly of the mitochondria increases. And this is sort of called pathogenic heteroplasmy. Another way I think about it is in terms of Pac-Man. So imagine that ROS are the little dots, and then antioxidants are the little waka wakas, right? And so in a young cell, it's a pretty good balance. In aging, the balance gets a little bit perturbed, but then like in extreme aging or like super chronic sickness, it's game over. Like you can't counterbalance it. So dysfunction contributes to oxidative stress within the cell, and aging exacerbates both of these things. Mitochondrial transplantation is this phenomenon that's been described now for at least 20 years, by which cells can donate mitochondria to other cell types. And so it can happen through a couple different ways that are characterized. The first one is through the tunneling nanotube method, which is the cells kind of build like this bridge and they handshake and they share mitochondria this way. There's encapsulation where either through microvesicles that bleb from cells or through like the synthetic way, which is like lipid nanoparticles, we can get mitochondria into cells that way. And then just the free-floating uptake of mitochondria also occurs. And like I said, it's been happening. This was first, I think, characterized, or at least the term of mitochondrial transplantation was first described in like 1930. So it's been increasing field of interest because of the potentials. And so when I started working on this, it wasn't quite bioengineering. It was really just like low down and dirty, using a centrifuge to deliver mitochondria to T cells, because I was interested in how we could improve immune function. And so particularly we were looking at CD4 T cells in context of aging and tuberculosis infection. And so just to show that it's simple enough to use a centrifuge to deliver mitochondria, we had some GFP tagged mitochondria. And then we did this procedure, and then we visualize the cells by confocal microscopy. And so on the top are the cells that are your control, and then on the bottom are the GFP tagged mitochondria after centrifugation inside your CD4 T cells. So it's a very simple process to get mitochondria into cells. And really just by augmenting the amount of cell type versus the amount of mitochondria that you have in your suspension, you can pretty much like get 100% efficiency more or less. And so this is just an example of that. And so we came up with a simple protocol where we then would isolate mitochondria from healthy fibroblasts and then mix them with the T cells for like about 5 minutes in the centrifuge, spin them, and then look to see what were some of the functional consequences of the transplantation. And so I'll just bring back this sort of perpetual loop of oxidative stress and mitochondrial dysfunction. And people that are not familiar with the seahorse assay, it's a glorified oxygen sensor and a pH like meter. So it detects how much oxygen is consumed by cells, and then how much the pH tends to give an indication of the rates of glycolysis. And so when we looked at the impact of mitochondrial transplantation in CD4 T cells as compared to young and then the old plus mito, we saw that the old plus mito group after transplantation had a significant increase in oxygen utilization in all the parameters. And these I'm just showing some for brevity, but it's all published. So you can either read it or have your AI read it to you, whichever one you want to do. But we saw that there was improvements in oxygen utilization. When we looked at ROS production, particularly mitochondrial superoxide production, this technique is called the resonance. And essentially the little squiggles are the peaks. The higher the peak, the higher the intensity of mitochondrial superoxide detected. And so we're showing that compared to young, old CD4 T cells produce a lot more mitochondrial superoxide. And then after mitochondrial transplantation, we see that the peaks decrease. And so there's a decrease in the functional levels of mitochondrial ROS that are being produced after transplantation. So even though there's an increase in oxygen consumption, there's a decrease in the amount of superoxide that's being produced. So it suggests that there's better efficiency of ATP production. We took some of these samples, some of the cells, and we did proteomic analysis on the cells. And gene ontology or goal ontology term analysis, we were able to identify some of the top upregulated functional changes, and a lot of them, the top two were related to the mycos and cristae, so this the mitochondrial architecture. And so we followed this up by doing transmission electron microscopy to compare the architecture of the mitochondria in the CD4 T cells that didn't get any transplantation versus the ones that did. And you can see that there's a very clear difference. I call this like the abs of the mitochondria look like the cristae are like abs right now because they're very protruding and very pronounced. And so again, corroborating that the changes that we see from the proteomics is also what we see in the TEM, but also a denser cristae architecture also suggests better utilization of the TCM metabolites or intermediates that mitochondria use for OXPHOS. And so all the data sort of corroborated that there is improved functionality. You got a Hemi in your Toyota Corolla. It's a better engine. And so typically when you compare CD4 T cell from a young individual or healthy individual compared to like an old individual that's responding to the same cognate antigen, typically the older T cell will display lower markers of activation. They'll produce lower amounts of cytokines with a bias towards pro-inflammatory cytokines, and they don't proliferate to the same magnitude as the younger T cell. And so we wondered what would happen if we could if we use the system to now deliver mitochondria to HT cells, how would that impact their ability to activate, produce cytokines, and proliferate? And so we looked at T cell activation. So CD69 and CD25 are markers of activation of T cells, and these are paired comparisons. So it's like literally cells from one mouse split into two groups, comparing with or without the mitochondrial transplantation after activation. And we saw that the T cell activation went up. We looked at IL-2, which is a cytokine super important for T cell proliferation and tends to go down with aging. And we saw that this cytokine, the production of this cytokine increased after transplantation as well. And so it kind of led us to also predict that there was going to be an increase in the ability of the T cells to proliferate. And that's what we saw. And these are all, at this point, ex vivo experiments. So it's all done outside of the mice. But the true question, or the true challenge, was to now take this and put this into mice and actually see if there's like a functional benefit to doing a transplantation just to the CD4 T cells. And so we took aged and naive CD4 T cells, and some got the centrifugation-based mitochondrial transplantation, and then some just got just vibes, just chilling. And we put them into RAG knockout mice, and these mice don't have an adaptive immune response. They lack an adaptive immune response. And then we challenged the mice with pathogens that we know requires some arm of adaptive immunity for either survival or lowering like bacterial burden. And so for influenza, we looked at the mortality rate of the mice after infection. And then for tuberculosis, which is one of the pathogens I studied, we looked at the CFU, or the colony forming unit burden in lung and spleen, because that's where it tends to go after dissemination. For influenza, what we saw was that so we had a group of mice that didn't get any CD4 T cells, a group of mice that got old naive CD4 T cells, and then the old naive plus the mitochantransplantation. And we saw that for the survival against the influenza challenge, the mice that received the old plus mito T cells, they had the best survival. When we looked at the MTB burden, again, different experiments, but when we looked at the CFU burden in the lung and spleen of mice, the mice that got the O plus mito T cells, they had the lowest mycobacterial burden, suggesting that there's a physiological benefit towards doing this transplantation. And then in vivo, there's a functional benefit towards it. In aging, there's like two sort of end phase that you get with your T cells or with your adaptive immune system. It's like they either become exhausted or they head towards like senescence. In tuberculosis, exhaustion, the longer you're infected with the pathogen, the higher the burden of exhaustion that you get. Well, you also get senescence too, but I'm not trying to confuse you guys. And so from our proteomic data set, we actually screened for proteins associated with T cell exhaustion. And so on the left here are the OC4 T cells, and then here are the OC4 T cells after mitochondrial transplantation. And we saw there's a shift. I'm not going to characterize everything that changed, but this was enough to convince us that we should characterize whether this is the same thing that we would see in vivo. And so we did another experiment where we took, this time, TCR knockout mice, because they tend to fare a little longer for these long-term experiments. And we either, again, gave them the old plus mito or like just the regular naive CD4 T cells, and then looked at we phenotyped the T cells at 30 and 60 days post-transplantation and post-infection with tuberculosis. What we noticed was that in the lung and spleen compartment, markers of exhaustion on CD4 T cells were lowest in the old plus mito group. And in this experiment, we had some TCR, so some TCR knockout mice that also got just young, naive CD4 T cells. And so the data is kind of suggesting that compared to even young CD4 T cells, the O plus mito group is actually working superior. So we went ahead and we did another experiment to sort of corroborate this using elderly CD4 T cells. The average age range was 60 years old. And for this to sort of mimic T cell exhaustion, we just chronically stimulated them for 7 days continuously. And then we phenotyped at 7 days. And so when we looked at T cell activation, again, we saw improvements compared to the LDC4 T cells that didn't get anything. When we looked at markers of exhaustion, we saw that these markers went down. So again, corroborating that mitochondrial transplantation diminishes T cell exhaustion in both mice and then in the LDC4 T cells. The other end fate of T cells in aging and chronic infections is senescence. And so again, we screened our proteomic data set for proteins associated with senescence, and we also saw that there was a shift. And again, we followed this up with an experiment looking at elderly CD4 T cells where we stimulated them, just a single stimulation for typically 3 days. That's what you could do. And then we phenotyped for markers of senescence like CD57, P16, and beta-gal. And so again, with activation, the O plus mito elderly CD4 T cells had better activation, lower markers of senescence, and then lower markers of senescence, so CD57, and then beta-gal P16. So again, suggesting that mitochondrial transplantation makes the T cells a little bit more resilient to exhaustion and senescence. So just to recap everything I've talked about so far, in case I'm going a little too fast, so mitochondrial transplantation in aged CD4 T cells increased OXPHOS. It reduced mitochondrial ROS that was being produced, and it had a drastic change in the cristae architecture, cristae density. It improved the activation of the T cells. It significantly improved the production of cytokines. I showed you IL-2, but some other cytokines also improved, and also improved like T cell proliferation. And then when we put these sort of rejuvenated T cells back inside in mice, it extended the survival of mice that were challenged with influenza, and it also reduced the mycobacterial burden in MTB infected mice, and also reduced markers of T cell exhaustion and T cell senescence. So if you're like, I did this experiment and I was like, bro, like, this is exciting. This is very simple. I mean, it really doesn't cost much. It's literally just growing cells and isolating the mitochondria and then spinning them. So like I could put mitochondria into a lot of different things. I could put them into macrophages, into T cells, vascular stem cells. My research is really geared towards the immune cardiovascular axis now. And so I think there's a simple idea that we could use on a lot of different cells, regardless of what the underlying role of mitochondria is within the cell. And so I'll show you another example of this principle in the macrophages. So back in the day, well, I don't say back in the day, but like about 10 years ago now, there was a paper that came out that talked about inflammatory macrophages and how they have issues polarizing, going from an inflammatory phenotype to anti-inflammatory phenotype due to mitochondrial dysfunction. And another interesting thing about macrophages is that if you were to put them under the microscope, like confocal microscopy, and visualize their mitochondria network, the M1 macrophages, which are the pro-inflammatory ones, they have a much more fused or hyperfused network compared to non-polarized or the anti-inflammatory like M2 macrophages. And so the question that we first wanted to see was, well, if we delivered mitochondria to M0 macrophages and tried to polarize them towards either inflammatory or anti-inflammatory cell type, what would happen? And so that's what we did. We did mitochondrial transplantation using the centrifugation method, and then polarized the macrophages towards the M2, and then looked at M1 looking at INOS and M2 looking at ARG1 as sort of proxies for polarization. And then the readout was by flow cytometry. And so here you can see we did like a sort of a dosing, different volumes of mitochondria to a consistent amount of macrophages. And so we see that towards the inflammatory or M1 phenotype, we did not, like, no matter how much mitochondria we added, we really didn't see an uptick in the fold change or induction of M1 INOS. But towards ARG1 expression, we saw that there was a dose-dependent increase in those genes. And so the question then became, could we then just give mitochondria to inflammatory macrophages, and would that actually help? Oh, well, we also looked at some other markers of M2 fate. CD206 is highly expressed on M2, and then ARG1 again. But then the question became, if we have these sort of M1 polarized macrophages, can we use mitochondrial transplantation to then make them more anti-inflammatory or less inflammatory? And this is the kind of experiment that we did. And again, we used intracellular staining to sort of, as a quick readout, and these are ongoing experiments. But so when we looked at ARG1 expression in both the groups of macrophages, M1 polarized that received mitochondria or that didn't, the ones that didn't are in gray, and then the ones that did are in red, we saw that the ARG1 expression across time was going up. But when we looked at INOS expression across time, we saw that in the mice in the macrophages that received mitochondrial transplantation, their inflammatory INOS gene or protein expression was going down. And if you put it on a ratio of ARG1 to INOS expression, we see that there's a better sort of like repolarization towards M2 or anti-inflammatory macrophages. And so we think that this is something that's simple enough that we can now manipulate and try to scale. And we're looking to see if we can do this in some cardiovascular models, because M1 macrophages or inflammatory macrophages are highly pervasive in cardiovascular disease. Now, before I was literally when I started doing this stuff, I was like, I can just put mitochondria into all these different cell types. But there's just a there's an issue I would say that became identified as I was doing this, and it's just like in the process of mitochondrial transplantation, in most of the literature that's published, it's just kind of like low efficiency. So in some papers here, like this one using the sort of the handshake method, it's about 10% internalization or transfer efficiency. And in this other paper, you got like 40%. Now, although using the centrifugation method, we could sort of augment and get almost 100% of transplantation. This method is not practical for cells that you cannot take out of the body, like endothelial cells. So this is like one of the cruxes, and I would say one of the major things in the field that needs to still be solved. And so, for like I said, the endothelial system in cardiovascular disease across all cardiovascular disease. Endothelial dysfunction is common, and mitochondrial dysfunction is an underlying symptom and consequence of cardiovascular disease. And so, because it's really not practical to take out endothelial cells and do sort of a centrifugation method of mitochondrial delivery, we have to think about a different way of getting mitochondria to inside the body with targeting. That's the keyword there, targeting. And so, to summarize, current methods of transplantation right now are kind of like a bus system where for in vivo delivery, you'll just inject mitochondria into an organism or a human and then just kind of hope that the bus stops at the right location, if you're lucky. Um, Instead, we wanted to come up with like something like an Uber approach where we could actually tell the mitochondria where to go. And so we took some cues from cancer biology. So antibody-drug conjugates, they work really well because the antibody is targeted towards a receptor typically on a cancer cell that then induces some sort of reaction, apoptosis or some other form of cell death. And so we thought about whether we could do the same thing for mitochondria. Why not just attach an antibody to the outside, and maybe we can actually have some targeted system this way where the mitochondria would preferentially be uptaken by cells that are expressing this receptor. And so I'm calling these things mitotags until I find a better name for them. And so again, similar to centrifugation, I like simplicity, but we came up with a simple method where we could culture donor cells with sprinkle some seasoning on it. We'll call it that for now. And then we isolated the mitochondria, and then we're able to really complex them with antibodies and to make these mitotags. And you're probably thinking, okay, well, just because you can get them into the cell doesn't mean like they're actually staying in the cell. Maybe they're just getting degraded or something like that. And that's the same thought that I had, like what are the potential fates of the mitochondria after internalization? Elimination is a possibility. Elimination of the native mitochondria is also a possibility. So donor versus native, but also maybe fusion of the delivered mitochondria with the native mitochondria, and then potentially full replacement or retention of the delivered mitochondria. And while we're still answering these questions, I think the data that I'm sharing sort of gives a hint as to what's happening. And so for these experiments, what we did is we would compare unmodified, or I call them naked mitos, versus the mitotags. And in the specific system, we used endothelial cells. And so we made endothelial cells highly express CD31, PCAM, that's the name for it. And so we made an anti-CD31 mitochondrial conjugate, or anti-CD1 mitotag, and delivered this. Well, because their antibodies you can make them fluorescent. We also had a fluorescent tag on it. And so we delivered these to endothelial cells to see if they would actually be internalized. And what we saw by confocal, so all of these red dots are indicative of a mitochondria or a mitotag that gets internalized into the endothelial cell. And so V-cadherin is another surface marker to sort of just show that they're inside the endothelial cell and not on the outside. So the next question became, okay, how much better is this sort of system compared to the unmodified or naked unmodified or naked mitochondria? And so we just, again, compared using confocal microscopy and flow cytometry to see which one worked better. And so for these experiments by confocal, again, because we fluorescently labeled the mitochondria, and then we just compared literally how many mitochondria went into endothelial cells co-stained with mitochondrial bread in the naked versus the mitotag system. And so if we're to quantify it, we can see that at sort of the low level, it was about, I think, a 10-fold increase. And then the highest point was like around a 50-fold increase. We went back and sort of did this experiment using flow cytometry to get much more quantitative readouts. And again, using sort of like a mitochondrial dosing volume. And we compared the unmodified versus the mitotag system. And we can see across all doses that we can scale this and we can get almost 100% deposition. And again, because we can also increase the volume more, but just on this scale, we can see that almost 100% of the endothelial cells take up mitochondria this way. We're also trying to understand the integration or like colocalization. In some of our experiments so far, it's by using ImageJ and sort of a colocalization software, Jacob, it's about a 0.8 threshold of overlap. So more or less an 80% integration of the delivered mitochondria to the mitochondrial nectar that's like some sort of pre-stained with the mitochondrial deep red, which is promising. It's not 100, but it's 80% is pretty good, I think. To look at the specificity, what we've done so far is we've taken the CD31 or the endothelial cell specific mitoTAG, and in co-cultures of endothelial cells and with a cell type that does not express CD31, like vascular muscle cells, we just sprinkle them on and watch to see whether there'd be any sort of preferential uptake. And so after 24 hours, we obtain confocal images. And so you've got the endothelial cells here in gray, we've got the vascular muscle cells here in green, and you've got the mitotags preferentially localizing to the endothelial cells. So it looks as though, like, because we said that it's a targeted system, it looks to be very targeted. And then we also did another flow experiment to confirm that most of the delivered mitochondria are localizing to or being internalized by the endothelial cells. Now we are seeing some that are getting to the vascular muscle cells, but that could also be because of just the natural handshake, the tunnel method by which cells will share mitochondria. We did another experiment to look at the ability of the mitotags to reduce oxidative stress. And so in context of inflammation, of course, because it's one of the main things we're interested in. And so we stimulated endothelial cells with interferon gamma for about 24 hours and then exposed them to the mitotags. And then 24 hours later, looked at by flow cytometry at the overall levels of oxidative stress in the cells. And we could also compare this against naked or unmodified mitochondria to see which one was working better. And so on the far left, you've got sort of your cells, control cells, endothelial cells, not exposed to anything. Here you've got your cells just exposed to interferon gamma, and we see an increase in, so DCFD is a general ROS probe. It non-specifically detects all the ROS inside a cell. And so we see that there's an uptake after interferon gamma treatment, but then after mitochondrial transfer or using either naked, with the unmodified mitochondria, there wasn't much of a decline, but with the mitotax system, we see that there's a significant decline in the levels of interferon gamma induced oxidative stress. So it suggests that there's a functional benefit towards a functional advantage towards like using the mitotax system over unmodified mitochondria for treating inflammation. Again, this is right now still ex vivo. Another question and this more so get towards the idea of retention. So Rho cells or Rho zero cells are some cells that they don't have mitochondrial DNA, and so they have no functional OXPHOS. They require aerobic glycolysis for them to live, but they also require uridine supplementation for growth. And so we said, if we have these Rho zero endothelial cells, which have no functional mitochondria, if we were to deliver the mitotags to these cells, would they then be able to have self-sustained proliferation? And our hypothesis was that, and we hypothesized that the sustained proliferation of the Rho zero cells without uridine supplementation would be possible with the mitoTAG system. And so here we have so in the gray are the Rho zero cells without any uridine. And you can see that there's a sharp decline in their proliferative capacity based off of EDU incorporation, which EDU incorporates into actively synthesized DNA, and you can detect it by fluorescent conjugation. And so we see that this goes down over time, but we see that with the mitoTAG system or the after mitochondrial transplantation, we see that there's somewhat sustained proliferation levels. And here we have Rho zero cells, the same cells from the gray, but in presence of uridine. So it seems as though, even though it's not like 2 months or something like that, at least right now, it looks like the mitochondrial transplantation through the mitoTAG system is sustaining proliferation of cells that did not have functional mitochondria. For me, I really think that this is just like the beginning of a pipeline. And that's why the title is like Bioengineering Mitochondria, because for the longest while, we were having issues identifying a scalable method to deliver ample amount of mitochondria to cells to have, I would say, a true clinical or functional change that could be scaled. And so now because we already have tools like CRISPR-Cas9 and even mRNA technology, we can manipulate the nuclear mitochondrial genome to augment some of its capacities, but also we can potentially introduce synthetic mitochondrial DNA into extracellular mitochondria and then package them and deliver them to cells to see how that really influences the cell's bioenergetics and ultimately the programming. Because I don't know of a disease, literally any disease right now, that isn't impacted by mitochondria or doesn't lead to mitochondrial dysfunction. So literally, this is a potential like a pipeline for a lot of conditions. And so anything with mitochondrial dysfunction is a potential area of interest. So neurodegenerative disease, of course, primary mitochondrial diseases, sarcopenia, ovarian aging, stem cell exhaustion, etc. I'm really interested in the immune system and immune aging. And because we had shown all this data, published all this paper showing that using centrifugation-based mitochondrial transplantation, you could reduce a lot of the markers of exhaustion and senescence in mice and in elderly T cells. We wonder if we could then make a mitotac for the immune system that is just targeted towards the immune system. And so CD11A is another receptor that on immune cells that undergoes endocytosis. So that's another property that's important. Like we select receptors that are known to do endocytosis. And so we made CD11A mitotac for immune cells. And again, we did sort of like a dosing comparison of the uptake of the naked or unmodified mitochondria versus the mitotac system. And so we see here, even at the lowest concentration, we can almost get like 80%. And again, it scales to almost 100% uptake. And even increasing the volume or the amount of unmodified mitochondria still doesn't get you the same, even at our highest concentration, does not compare to the mitotac. So here's just a quantification of the fold change. So we're really now trying to, based off the funding that I've gotten for this, is really the repository of different mitotacs. So different receptors for different cell types, so we can have really targeted delivery. So if you could envision sort of like a cocktail for mitochondria for different cell types and all infused from one single dose, I think would be kind of cool. And so we're looking at generating different mitotags for the immune and cardiovascular cells and beyond, and then sort of evaluate their efficiency and then really customize and bioengineer with augmented properties, which I think is, it's the future. It's the present, if you let me keep doing this. So another project that we have is funded by American Heart Association, where we're actually going to use the mitotac system to see if we can alleviate aortic aneurysm formation in mice, especially because we have a mitotac that's specific to endothelial cells and we have a mitotac that's now specific to inflammatory macrophages, which I didn't show you in that data, but I'm surprised. And then there's another project that we have funded by NASA, a pilot project where we're essentially going to create mitotacs that can be they're going to be pre-made and sort of cryopreserved so that astronauts can take to space and potentially use as a way to, because in space travel, everyone wants to go to Mars, but y'all don't understand the amount of radiation damage that you're going to incur up there. So you need some way to really buffer yourself from that levels of radiation. So this system we're thinking of it as a way that you can have sort of like, if you were to do a spacewalk, or if there's like a solar flare or something that hits your spaceship, you can get your mitotags from storage and give yourself a boost. And this is my collaborator at NASA, Robert Hinshaw. Another project that we have going on right now is, again, because we made a mitotac for the immune system, is we're going to deliver these into aged marmosets and then see if there's any sort of quantitative readouts towards like systemic inflammation, immune cell sort of like frequency, whether the restoring mitochondrial functionality in the entire aged immune system, like in periphery, does that actually have a shift? Does it reduce inflammation? Then also does it lead to like a shift in the sort of frequencies of effector or memory or innate cells that are like running around? What else? Yeah, so these are things that I kind of wanted to highlight, and I'm happy to collaborate with people because I think that what ultimately is built here is a tool that I would like other researchers and other individuals to test out the mitotags in their own systems. So with that, I will take any questions.
Thank you so much. It's totally fascinating. I really want to hear more about the mitotags in space, if possible. But first, Micah, please go ahead.
I'm curious, what is the advantage of targeting specific cell types rather than just mass producing enough mitochondria to overcome the efficiency issues with naked injection? Is it just a cost thing, or is there something else that makes it bad to just inject huge volumes of naked mitochondria?
So in the context of aging or in the context of infection, your cells are not all they're not using all mitochondria at the same time, but they don't age at the same rate. And so it would make much more pragmatic sense to target tissues that are high oxphos or with high burdens of mitochondrial dysfunction as opposed to those that don't. But also because right now in the literature, there's evidence that, so for example, cancer cells can hijack mitochondria from healthy cells and then with T cells, they make them more exhaustive. So it would make more sense to have a targeted system because you can then target, you can preferentially give healthy mitochondria to cells that need them, but then also maybe give apoptotic or pre-programmed mitochondria for cell death to cancer cells. So I think it's a better way for that. It's a better system for that reason.
Awesome. Joel, please go ahead.
Yeah, thank you, CoCo. Really great presentation. Every time I listen to you, I learn so many new things. So one question is with respect to the macrophages, really interesting, and there's so much that can be addressed by turning those macrophages to an anti-inflammatory state. So just to make sure I understand, in order for this to happen, the hypothesis is that the mitochondria need to be internalized by those macrophages. So that's one question. And then if you can maybe expand a little bit more about just the fate of the mito tags once they're internalized. What do we know about how long do they last there, and etc. So thanks.
Okay, so let me see if I remember. So the first part of the question was towards like the reprogramming of the inflammatory macrophages towards like an anti-inflammatory phenotype. So the original paper that I saw, they used the sort of the co-incubation of dendritic cells with inflammatory macrophages to show that dendritic cells would donate their mitochondria to inflammatory macrophages and reduce their inflammatory or M1 phenotype. And so that paper suggested that they needed to be internalized. In experiments that we've done where we've used unmodified or like naked mitochondria, where we didn't do centrifugation, but we used the mitoTAG system, we saw that just incubating the macrophages with unmodified naked mitochondria did not increase their sort of like plasticity or M2 phenotype as well as the mitoTAG system. So it seems as though there needs to be internalization, one, but then also because I believe it's the lactolylation or it's a histone modification process that is dependent on active mitochondria functioning inside the macrophages. And with the M1 macrophages, it seems to be completely broken. Well, at least, I'd say largely broken. So yes, we need to have them internalized. And then the other question was, can I talk a little bit about like the sort of the dissociation of how long are the antibodies or like the attached moieties on the mitochondria sticking around?
Well, no, mitochondria with the tag is internalized. What do we know about how long does it stay there compared to the existing mitochondria, the native mitochondria of the cell?
Good question. We don't know enough yet. So most of the experiments have been with, so to do these experiments, we need typically two different of mitochondrial DNA to do tracing. Most of the experiments I've done so far have been in row zero cells just to show that there's retention. It seems that there is retention. And so the next question would be, for example, can I use the mitoTAG system to flush all of the native mitochondrial DNA out and replace it with donor mitochondrial DNA? The same way or the reverse method of how the 3-parent mitochondrial babies are made, where they do like denucleation. So this would be a much more efficient way to flush mitochondrial DNA out of cells. So check back in like 3-6 months, maybe have a better answer. Jason.
Yes, thank you, CoCo. That was a great presentation. I was just curious a little more on targeting different cells in the body and if you would need a different mitoTAG for every different cell type. And say if you had a cell type, say you were looking at like skeletal muscle, for example, and you had a mitoTAG and you put it into an IV, went into a human, at that point, I mean, would the immune system let it slide through, or would the immune system sort of think, oh, this mitoTAG is foreign, let me remove that from the body? Like, I was curious how you're kind of going about these problems.
I mean, they're antibodies. So I don't expect the immune system to necessarily go crazy over antibodies because we inject them all the time. healthy mitochondria don't seem to induce any sort of inflammatory response. And so it comes down to sort of the quality control of how you prepare the mitochondria so that they're not inducing any sort of inflammation because of just inherent damage. And towards the question of, would you need to have a different mitotype for every different cell type? And technically, no, because there are receptors that we know do endocytosis on all cell types, like the LDL receptors, like one of those that you can easily use as a target. That if you wanted to sort of target everything at the same time, what I'm trying to demonstrate is just that if you wanted to do that, you could, but there's also selectivity, especially for cases where we know, I guess the prime example is with cancer, where it might hijack healthy mitochondria. So we want to make sure that we're sort of prioritizing delivery, like I guess Amazon to some extent, like getting your shipment on time. So
I loved the Uber example. It was great. So if you were to like bioengineer these, you would just be taking a part out of the body, like it would be all self. Like you wouldn't have like a donor or would you need a donor, or would it be just from the same human?
good question. It could be both. That's the sort of beauty of this, because so for me, at least how I view the bottleneck in the space of transplantation has always been just scaling. If you wanted to take cells from yourself and then enrich them for healthy mitochondria and then repurpose them and add a mitotype to replenish yourself, you could do that. But also we're looking at sort of allogeneic applications, and we need to better understand the sort of the integration of mitochondria, like donor mitochondrial DNA versus like the heteroplasmy that would be incurred from allogeneic transplantation, and if it would actually be sort of like a sustained phenotype. So I don't know if that completely answers your question, but
No, I think it does. That's good. Yeah, thanks, CoCo. I really appreciate it.
thank you.
Thank you.
Micah?
We can let Lynn go first. I already asked something. I'll come back to me.
It looks like Lynn is up next.
Hi, has this approach been tried in long COVID or chronic fatigue?
It has not been tried yet, but this is why we're kind of doing the marmoset study, because if we show that there's a decrease in systemic inflammation in aged marmosets, then I'm pretty sure that it's going to be an easier sell for any sort of chronic immune condition. But also, one thing I'm also interested in trying is just something like CAR T cells. Can we, because we know that they're prone to exhaustion, can we just some mitotags and see what would happen if that would actually boost their anti-exhaustive potential? So
Excellent. Thanks for your work. It's
Thank you.
fascinating.
Micah?
Yeah, I wonder if you could comment at all on the cost and complexity of the tagging process and how that compares to the cost and complexity of actually just manufacturing all the mitochondria. Like, is the tagging relatively cheap compared to the cost of producing mitochondria, or is the tagging the expensive part of the process?
I mean, producing mitochondria, you really just need some media. So you need some media and some petri dishes. So it's easy enough to scale fibroblasts or a cell type that has a decent mitochondrial content. And now there are protocols that you can actually augment the amount of mitochondria within a cell. So I would say just based off of that, it is cheaper to just grow your cells. The making the mitotag, or like the antibodies, are expensive. Well, and depending on like how you process it can be expensive. But compared to as far as scaling, because it literally occurs in 3 to 4 steps, I don't think it's going to be an issue price-wise as we sort of increase the amount of mitotags that we make.
Okay. And last question. Well, during your presentation, you mentioned you might want to enhance mitochondria before putting them in. I'm just curious what your thoughts are on what are some enhancements one might want to make to their mitochondria. Just pie in the sky, what are you imagining? What would you do to make better mitochondria?
So, for example, this is all I've been thinking about. Well, okay, there's two potentials that I think. One is great, and then the other is like, I'm scared. I'll start with the scared one first. Blood doping. I think like doping in general, like, bro, people are going to start, if this really is not like properly controlled, I think people are going to go crazy with the potentials of augmenting your own ability to run faster and stuff like that. So that's one thing. With every good thing, I think there's a bad application. One thing I've thought about towards in the cardiovascular space is there's a lot of, in general, typically you have a lot of lipid buildup. And we know that our brown adipose tissue, we have these mitochondria that have the UCP1 expression on them. And so I've been thinking about really engineering mitotags that have an overexpression of UCP1 and then delivering them to foaming macrophages to see if that reverts because foaming macrophages are inflammatory. So would that actually help with reducing systemic inflammation through that method. So that's one potential augmentation. But also you think about there isn't a process inside a cell that does not sort of, it isn't like brokered by mitochondrial. And so I've thought about sort of, what's the word, augmenting the calcium buffering potential of mitochondria to see that then how would that lead towards like better signaling or again, changing the instances of exhaustion or activation and stuff like that. So that's just towards like the immune system. But then you think about the brain, maybe you can insert mitochondrial non-native genes into the mitochondrial DNA and then deliver that and have that as sort of like an augmented expression. So we can have a long conversation about this stuff clearly, but yeah.
Yeah, very cool. There's a group trying to make it so mitochondria can generate ATP from light instead of needing oxygen. And so you could
you're talking about Shahaf.
Yep.
Yeah, I know him.
Yeah, exactly. So,
he's a friend.
So we could maybe do something like that would also potentially work here. It would be very cool.
Yeah.
Okay, thank you very much.
Yeah. Robert? I think you muted.
You muted, Robert.
Sorry about that. First, nice work. Looks like you've been very busy. Before I forget, I wanted to mention on terms of looking for sources of mitochondria, one of the places that I think is very interesting is to look at oocytes. Oocytes generate huge numbers of mitochondria, and the inherent mechanisms that are activated inside of oocytes involve some of the best quality control of any cells in the body too. So just something I thought I'd mention. And then I just wanted to ask about the endothelial cells that you're using. Is that a cell line? Are those primary?
Primary. Yeah, it's all primary.
Okay. All right. That
you took for the oocyte. Yeah.
I remain skeptical of things until they happen in vivo, but better that they're at least the primary cells and not a cell line.
Oh, check back. So, like I said, the age marmoset study will, I guess, make or break the claim, but check back in maybe 6 months. We'll have some data.
Excellent. Thank you.
Thanks.
Awesome. We're just about at time. However, if there are any last burning questions, speak now or forever hold your peace. Anton, would you like to ask the question, or I can always read from?
I'm just curious if like mitotags are so efficient that for some cells it will like get a huge number of mitochondria, more than it typically has according to its cell type. What can be potential cell behavioral changes?
So, I mean, typically, when we've seen this with just other methods of transplantation, if you give a cell too much mitochondria, they'll just degrade them. So I don't think, at least right now, I don't see that as a major issue. Now, if you overload a cell with like exuberant amounts of mitotags, you might induce like oxidative stress just because you're literally overexhausting the cell towards like using oxygen. And just by proxy, you're producing more superoxide if you look at the mat then. So in most of my experiments, I'm trying not to overload them. So.
Amazing. Well,
you had a question towards the space. I'll see if I can. So what was the question again? Like, what do you want to know? Like, what specifically about it? Or Lydia, you?
Oh, I saw your NASA slide about the application of mitotags in space with decreasing radiation.
Mm-hmm.
And I just want to know more about it. It sounds so cool.
I mean, look, I don't want to say I like my science spicy, but I don't believe in impossible. And I think from everything I've heard from NASA's side, shielding technology towards like space travel is not necessarily working as well as they expected it to go. And so knowing that there's been a couple papers now, the SOMA package included, that talks about mitochondrial dysfunction, multi-organ mitochondrial dysfunction being like a major signature of long-term space travel. We just figured instead of trying to dose with antioxidants, because that doesn't actually fix underlying issues with mitochondrial DNA mutations and whatnot, we just said, like, if we can at least supplement an astronaut with healthier mitochondria, maybe that decreases the overall burden of radiation damage that they accumulate. And we're doing experiments to really test that out. We're essentially seeing if we can cryopreserve the mitotags, and then after reconstituting them, can we then give them to immune cells that have been irradiated, and does that actually reduce their exhaustion or senescence markers after they've been sort of activated or stimulated? So I think there's a potential here. So I'll keep you posted.
That's awesome. That could be huge. Well, I think we're at time. So thank you so much, CoCo, for joining us. Finally. It's been such a pleasure to have you on the group. Thank you, everyone, for your attention and your great questions. And also, just a note that if you joined via Luma and you liked this a lot, you can email me at lydia@foresight, and we could get you onboarded if you want to come back. But thanks again, CoCo, and we'll see you all again next month.
All right, it's been a pleasure.
Thank you so much. Have a great day, everyone.